11B. There, it's all better. Sorry. All right, so 11B is nice. It's a little lighter than what we just saw with the transcription, the translation, DNA replication. But we do need to apply what we've learned in this second half of chapter 11. Because we're going to look at two concepts. We're going to look at mutation as it applies to prokaryotes and to some extent eukaryotes. And then we'll get into newer concepts that most folks haven't seen in Gen Bio. Horizontal gene transfer. Something very common to bacteria and something that's actually causing humans a lot of problems. Okay, so when you get to the genetics units in Gen Bio, a big part of it is talking about evolution. Usually it's like three separate chapters that address evolution. Such a big concept. And evolution is the gradual change in organisms over time. Okay? Organisms evolve and adapt to changing environments over millions, hundreds of millions, or billions of years. Now, when you talk about this in Gen Bio, they get into the different mechanisms behind what drives evolution. Okay, and you spend entirely too much time talking about natural selection, kind of the biggest force behind evolution. And as a reminder, if you've not talked about it, natural selection is the hypothesis that given a population of organisms, go in the forest, say you got a bunch of deer. The most fit organisms, the biggest, baddest deer, will be the most successful at reproducing, at passing on their genetic information. Okay, that's natural selection. We naturally select for the organisms that are best suited to reproduce and pass on those successful genes. And, in Gen Bio, they use great examples like big bad deer, big bad bear, whatever. They don't talk about bacteria, though. Natural selection also applies to bacteria. And, one of the hard things to grasp with evolution is, these changes take a long time to happen, typically. Not for bacteria. Okay, you can see selection happening in much shorter periods of time. So, for instance, when I was in grad school, we did what was called metabolic engineering. We used to engineer E. coli to eat sugars and poop out valuable alcohol. The problem is that E. coli can't handle a lot of sugar, maybe 2% tops. And we wanted this guy to make cash for us. We wanted to crank out as much alcohol as possible, so we needed to up the sugar. So how did we do that? Well, as far as I know, it's still going on, but every day, for the 6 years that I was there, we took that E. coli culture, and we threw it in a new growth medium with just a little bit more sugar. So over a 6-year period, we went from 2% sugar to 11%. There is not a gram-negative on the planet that can handle that sugar concentration and still live. But we had one. Because we, albeit not naturally, we artificially selected for E. coli cells that could gradually handle more and more sugar over time. And again, that was evolution in a 6-year span. There's this crazy dude, I always forget his name. He did a similar experiment with our friend Pseudomonas aeruginosa. Pseudomonas aeruginosa can't use citrate. He did a 27-year experiment where he kept growing Pseudomonas and he kept trying to force feed it citrate every single day. And within 27 years, it evolved the ability to utilize that citrate. Okay, so gradually over time, it acquired the means to do so. How? Mutation. So in both examples I talked about, the driver behind the wheel of natural selection, evolution, whatever, is mutation. Small changes in your DNA over a period of time. Okay, so that's the first thing we'll look at in this chapter is mutation. Okay, how do permanent changes happen in an organism's DNA? Now, when we talk about these happy little accidents, okay, something to realize about mutation, they have to be stable and heritable. Because if you take genetics, you will find out there is such a thing as a lethal mutation. So a couple of examples. I always forget how it works out. Mexican hairless dogs. I can't remember. They're all born hairless because if you get one with the hair genes, it dies as a zygote. It's a lethal mutation. Okay, looking at Manx cats from the Isle of Man. Females can never be calico because if you produce a calico female, it dies as a zygote. It's a lethal mutation. So, for a mutation to stick, to be expressed, it must be heritable. You have to be able to pass it on to the next generation. Yeah, don't quote me on the Mexican hairless dogs. I always mix up if it's haired or hairless that dies, but forgive me. So, when we talk about mutations here, we will find out, and why this builds off of 11a, DNA polymerase is not perfect. Most mistakes are random, happy little accidents during replication. And then what we'll get into next time is how you can cause more mistakes to happen through mutagens. All right, now the second thing here, the second driving force behind bacterial evolution is gene transfer, or horizontal gene transfer. Typically, I find this is a topic nobody's ever encountered in the past. So this is kind of a neat concept. Take two bacteria. They can be totally unrelated. Bacteria A says, hey, I got some super useful genes. You want them? Bacteria B says, yeah, I'll take them. And then they trade. That's horizontal gene transfer in a nutshell. We'll discover this is super scary because bacteria A could have said, hey, I got genes for antibiotic resistance. You want them? Yeah. It really happens, and it's a huge problem. So next time we'll get into the three mechanisms of how bacteria swap DNA: conjugation, transduction, transformation. All right, putting it into prettier pictures here. So when mutations happen, again, typically they're random accidents. But when that random mutation happens, if it's stable and heritable, it gets passed down through the generations. Okay, sometimes we call it vertical gene transfer. Now, with horizontal gene transfer, okay, take two unrelated cells. Okay, say cell A's got, hey, I got this neat plasmid, you want it? Cell B says, sure. And then they transfer plasmids. But, when this happens, this guy acquired new DNA. So that's technically a mutation, right? It sure is. Okay, so getting into mutations, what causes them, and ultimately how do we fix them, but we'll talk about that next time. So mutations are just changes in an organism's DNA sequence. And again, like we said, they must be stable and heritable, because if not, the cell dies. So a term that they talked about to death in GenBio is genotype. And for many folks, that sends shivers down your spine because when you deal with eukaryotic genotype, you talk about things like alleles. Okay, humans have 23 pairs of chromosomes. 23 from mom, 23 from dad. Okay, so you have backup copies of every gene. Okay, those are alleles. Bacteria don't have alleles, folks. Remember, they all have how many chromosomes? One. So no alleles, just one chromosome to worry about. Also, good news. In GenBio, you have to talk about Punnett squares and homozygous dominant, homozygous recessive, heterozygous. None of that applies here. Prokaryotic genetics is way more straightforward. Okay, so when mutations happen in bacteria, it does change their genotype. Okay, because they got one chromosome and one set of those genes. So if a change happens in a gene, their genotype changes. Now, when a mutation happens, you obviously call the organism after the point a mutant, right? Because it has a mutation. Now, another fun term I find they don't talk about much in Gen Bio is, what do you call the organism before the mutation? The wild type. Now, I want you to remember this term because wild type is a very common term used in genetic modification. So you got GMO everything these days, right? The GMO organism is the mutant. The template organism prior was the wild type, which we'll talk about more in the next chapter. All right, so when mutations happen, when changes happen, it typically changes a bacteria's genotype. Now, phenotype. Right? Phenotype is your outwards characteristics. Hair color, eye color, attached or detached earlobes. Do your ring finger and middle finger tilt in or are they flat? Okay, that's your phenotypes. Now, what about bacterial phenotypes? They don't have hair, they don't have eyes, they don't have fingers. What do they do have? Enzymes, biochemistry. Everything we're doing in lab right now, catalase, oxidase, nitrate reduction, those are phenotypic tests. Right? E. coli's catalase positive, oxidase negative, nitrate positive, that's its phenotype. Okay, so just to put that into perspective. But here's an important thing. Let's take a gene, any gene. On average, they're what? 1,000 nucleotides. If you change one nucleotide out of that 1,000, do you think it'll make much of a difference? Well, I'm totally baiting you because the answer is maybe, maybe not. Okay, odds are changing one nucleotide out of 1,000 isn't going to change a thing. Mutations typically have to accumulate over many generations. You know, changing one nucleotide, probably won't change anything. But a bunch of generations later, when now there's like 30 changes to that nucleotide sequence, well then, yeah, maybe you're going to start seeing some differences. So that's an important thing. Don't necessarily think that one tiny mutation is going to change anything. It could, if it hits just the right spot, but typically it takes time. Many generations of many changes. All right, so like I said, most mutations are purely spontaneous. DNA polymerase is not perfect. It does leave the occasional mistake behind. Okay, so most mistakes happen during replication. But as we'll see, we can also induce mutations. We can force DNA polymerase to make more mistakes by exposing it to mutagens. Now, you may not be familiar with chemical mutagens, but I guarantee you're familiar with ultraviolet light and what it does to your skin. Okay, sunburns are radiation burns. It's damaged DNA to the point where your body says, well, those cells are garbage. Okay, so we'll talk about that next time. Okay, so what I want to talk about next, or I'm sorry, I need to pause it. Any questions about mutation in general, natural selection? Okay. All right, so there are two major mistakes that DNA polymerase can and will make. Okay, one is point mutations, the other is frame shifts. Now, DNA polymerase is going to make a mistake. It's going to be a point mutation. Okay, the vast majority of the time. This is its most common mistake. So, this is how it works. So imagine the leading strand. DNA polymerase is reading and copying that template strand at 1,000 nucleotides per second. It's cruising along, it's cruising along, it's cruising along, and then all of a sudden, it throws in an A instead of a G, and just keeps going. That's it. Innocent little mistake, right? You know, throwing one wrong nucleotide, no big deal. Depends. So another name for point mutations, which in my opinion makes more sense, is base substitution. Because that's exactly what it is, right? You substitute a different nitrogenous base, a different nucleotide. So I'm going to draw this on the board because I want to show you something the book doesn't do a good job of. How this happens and how many generations does it take? All right, so let's make up some arbitrary 5 nucleotide sequence. What do I want to do today? We'll do G. My G's are awful. I'm sorry. G C A C T. Whatever. So bottom strand's got to be complementary, right? C G T G A. Thank you. Good review, right? All right, so we're going to copy this. So let's say nothing bad happens to the top strand. So that guy stays normal. Oh, sorry. Proper annotation here. Wild type. G C A C T. Okay, so that's a template. So we make the new blue strand, and let's say we don't make any mistakes. That one's fun. C G T G A. Now, on the other hand, let's say we're going to make a mistake when we copy the blue strand. So the blue strand here is template, so that's okay. Can't make a mistake with something that's already there. So the mistake happens on the new strand alone. So let's do this. Let's do G, C, G, C, T. Okay, do GT pair? No. Okay, so this is how the mistake begins. It begins on only one of the 2 strands, the new strand. This is what they don't show you in the book. So that's the first generation. Now, if this gets copied, then you get the point mutation. Yeah, my girls have been watching too much If You Give a Mouse a Cookie, and they say if, then constantly in the show, and that just went through my head. I'm sorry. Totally unrelated. Come home and I hear, mouse cookie, mouse cookie. But anyways, I'm sorry. All right, so blue strand is A-OK, right? No changes there, so that remains wild type. Let's copy this top strand. All right, GCG CT. Okay, so mistake is right there. So if we copy that mistake, CGCGA. Now the mistake is on both strands. So this is what they don't show you well in the book. They just go from wild type to mutant. They don't show you that it takes 2 generations. The mistake starts on one strand to begin. And then if the mistake gets copied, then it ends up on both strands. That's your point mutation. Now, something we'll talk about next time is these can be fixed. There is a system for that. But we'll save that for next time. Okay, but folks, any questions on how I went from wild type to mutant? Okay. So this is how it establishes itself. Now what we really care about is what happens to the codon sequence. Say this happens in the middle of a gene. Okay? If it happens in a gene, it's going to change the codon sequence. And the ribosome doesn't know any better. Ribosome doesn't know when these happen, so it's going to read it and just use the genetic code to figure out what amino acid it needs. So there are 3 possible consequences of point mutations. Okay, so 3 possible changes to the codons. So if you change the codon sequence, but because the genetic code shows degeneracy and it's redundant, so if you change the codon sequence and the amino acid does not change, that's a silent mutation, right? There's really no effect. Now, missense mutation. You change the codon and you change the amino acid. Okay, will that affect the protein? Depends. If your average gene is 1,000 nucleotides, that means your average protein is about 300 amino acids. So you change 1 amino acid out of 300. Is that going to change much? Depends. It'd have to be a really important amino acid. Now, worst case scenario is nonsense or null mutations. This is where your codon changes to a stop codon. So imagine this happens right in the middle of the gene. Ribosome going to know any better? No. It's going to hit that stop codon halfway through, and what are you going to get? Half a protein. Garbage. These are the worst case scenario. All right, so putting it into prettier pictures again. But like I said, they just jump into the consequences. So looking at this set of 3, okay, normally that gene has the codon UGU, and you should get cysteine, which is actually a really important amino acid for proteins. Now, what we're going to mess with here are the nucleotides on the end of the codon there. So, say we change the TA pair to CG. That becomes UGC, and that still encodes for cysteine. So, silent. No major changes. Now, let's say we switch TA to GC. That's UGG, that's tryptophan. Missense, right? We change the amino acid, and honestly, that would probably affect the protein. Now, I bet you can guess what the last example is. Okay, super unlikely, but TA could flip-flop and become AT. That's UGA, right? UGA, one of the stop codons. So, nonsense mutation. All right, so any questions on the point mutations, or especially the consequences of point mutations? Okay, so again, no big deal, right? Changing one codon. Cystic fibrosis is the result of a single point mutation. So typically, not much of an effect, but you hit the right place, it can be very detrimental. All right, so that's the most common mistake. Now, this one won't take me as long. The other mistake DNA polymerase may make, and this one's pretty unlikely, but again, it does happen, is a frame shift mutation. So this is how this works. DNA polymerase is reading and copying, and all of a sudden it stutters. Now, instead of throwing in the wrong nucleotide, here, either it's going to add one or more nucleotides where there wasn't any, or it will remove nucleotides. So we're not swapping, we're adding or removing entire segments of DNA. This is bad. So we talked about translation. I talked about the importance of the start codon. Right? The start codon sets up the reading frame. This is why it's frame shift. If you add one nucleotide, all the other codons get pushed down by one and out of frame. If you add or remove 2 nucleotides, they all get pushed down by 2 nucleotides, still out of frame. So basically, from the point of the frameshift mutation on, becomes garbage. Okay, all the codons get pushed out of frame, all the amino acids change, you get something totally different from what you wanted. With one exception. If you add 1 or 2 nucleotides or remove, It's bad. But what if you were to add, say, 3 nucleotides accidentally? That wouldn't necessarily be so bad. Why would not adding 3 nucleotides be so bad? What are you adding with a codon? Yes, so if you add 3 nucleotides, you're adding 1 bonus codon. It's kind of like a missense mutation. You know, you add 1 amino acid, may or may not affect the protein, it depends. But that's really unlikely. All right, so in this example, this is our typical healthy gene. This is our normal set of codons, and you should get arginine, tyrosine, valine-lysine. But let's say we push them down by 1 at this point. So we had AT where there wasn't AT. Everything from that point down is shifted by 1 nucleotide. Look at what it does to the codon sequence. So the first 3 are okay. You still get arginine. But after that frame shift mutation, now you get isoleucine, arginine, and now there's a stop codon where there wasn't one before. Frame shift mutations ruin genes. Because everything from the point of that addition or deletion on is garbage. Now, I'm going to be really brief with this last one here, and I'll refresh over these guys next time. But this last quick example here, the weirdos, transposons, aka transposable elements, aka, I love this one, jumping genes, has nothing to do with DNA polymerase. They are their own thing. And textbooks all put them in different places, which is ironic. What's that? Okay, so imagine this. You get a segment of DNA, usually a few thousand nucleotides long, that says, I'm bored of being in this particular spot on the chromosome. I want to move to a different spot or a different chromosome. That's a transposon. Literally mobile DNA. It will loop itself out from one point, move, and insert itself in another. So skipping to the end here because we're out of time, this applies to mutations because what if it jumps into the middle of a gene? If you add 5,000 nucleotides to the middle of a pre-existing gene, you think that gene's okay? It's garbage. So we call it insertional inactivation. And scary thing to think about this weekend, or this weekend, it's not that late in the week. We've discovered the human genome is riddled with these things, but have nothing to do with DNA replication. Okay, they are their own thing. And a transposon is a segment of DNA that can move. And by move, I mean it can move from one point in a chromosome to an entirely different point in a chromosome. Okay, this is why they're sometimes called jumping genes, because they jump from chromosome to chromosome, from location to location. This is really watered down stuff. If you ever take molecular biology, there's a whole chapter on these guys. They're very complicated, but they can be somewhat short, but most are thousands of nucleotides long. Okay, within this stretch of DNA that is a transposon, they do carry genes that produce enzymes that allow them to move. Their own scissors and glue, if you will. Now, the big concern we're looking at here is what happens when the transposon moves. What if it inserts itself in the middle of a pre-existing gene? So take a gene, 1,000 nucleotides on average, and add several thousand nucleotides in the middle of that gene. Okay, it's going to ruin that gene, right? You just can't add a whole bunch of new DNA and expect the gene to be okay. So transposons can cause insertional inactivation. They insert themselves in genes, inactivating them. Now, the neat thing to consider is what happens when the transposon moves again. The gene can come back together and reactivate. But that's a big if. So This is the diagram you typically see in books. It's not the greatest. But here they show a transposon that wants to move from one chromosome to another. Okay, here it does and shoves itself in the middle of gene X. OpenStax actually has a somewhat simple, realistic version of what happens. So here's a transposon. Here's the gene it wants to target. Typically, the DNA actually gets folded over so the transposon and the gene align. And then the enzymes involved, transposase, of course, will insert the transposon in the middle of gene B in this case. Okay, but gene B has been inactivated once that transposon is inserted. I bring these up because they are more common than we ever thought. Prior to the Human Genome Project, we thought these guys were kind of rare. Turns out that's not the case. Your DNA is riddled with transposons that got stuck once upon an evolutionary time. Okay, so that's it for all the spontaneous mutations, but any questions about the stuff we went over last time, or transposons? All right. So how often do these mistakes happen? So DNA polymerase makes a mistake about once in every billion nucleotides. So that's pretty impressive when you consider the fact that it reads at 1,000 nucleotides per second. Okay, that's pretty good accuracy there. Now, the problem with averages in statistics is that average doesn't always tell the whole story. Okay, sometimes range tells a better story, or median tells a better story. So the range of mutations is somewhere between 1 to 1,000 to 1 to 1 trillion. Because, as we've learned, some genes, some areas of DNA are hotspots for mutation. They happen very often. So, for instance, the genes for your antibodies are subject to a lot of mutation because your immune system has to adapt to infection. And part of the way it adapts is by purposefully mutating those antibody genes so that they can become more customized. Now, other regions see almost no mutation. So the genes for your ribosomes, for rRNA, they rarely see mutation. Okay, they're very short sequences, and when you screw with one nucleotide, you could ruin the entire ribosome. Okay, so those areas tend to be very conserved. They see very little mutation, maybe 1 in a trillion nucleotides read. All right, but what we're going to talk about next is how to induce mutations. How do you force DNA polymerase to make more mistakes? Okay, and we're going to do that by exposing cells to mutagens. Okay, mutagens can be chemicals or they can be physical, like radiation. And we want to alter the DNA or damage the DNA in some way that we force the number of mistakes to go up, the number of mutations to rise. Okay, so looking at some examples of chemical mutagens. Okay, chemical mutagens tend to either modify or replace nucleotides. So most of them, and this is all they do, most chemical mutagens just alter the base pairing rules. Right? If you mess with the high holy base pairing rules, that's probably going to start causing mutations. All right, so for instance, nitrous acid. Nitrous acid is a known chemical mutagen. It targets adenine. And it modifies adenine, and you don't have to know this, but it changes one of the functional groups. So here's adenine. Here's an amino group. Here it changes to a ketone, I think, or aldehyde. I always forget. Now the problem is that modified adenine, it doesn't just base pair with T, because right, it's A with T. This can base pair with C. So you can get AC pairing. That's going to cause some problems. So in the example at the bottom, okay, sorry, I took this from a different text because yours doesn't have good examples of this. But here's your wild type DNA. Say that this adenine gets modified from nitrous acid. If DNA polymerase reads this modified adenine, look what could happen. Now you got an AC pair. Now, if that cytosine gets read again, look what happens. Now it's a CG pair. So we started with TA, now we have CG. So this caused a point mutation. Okay, so nitrous acid modifying nucleotides will cause point mutations. All right, now base analogs, another example. These are chemical mutagens that look just like nucleotides, but they have different base pairing rules. Okay, so these guys can replace nucleotides in a very negative way. So for instance, at the top, we have adenine again, top left. Its analog is 2-aminopurine. 2-aminopurine can base pair with either T or C. So that is also going to lead to point mutations because this can trick DNA polymerase into throwing in the wrong nucleotide. Thymine, bottom left, its analog, and this is a common one, 5-bromouracil. Okay, 5-bromouracil can base pair with A or G. So this also can lead to point mutations. Okay, so here, most chemical mutagens just mess with the base pairing rules. Okay, so that you can cause point mutations. Now, something even worse is intercalating agents. These guys cause frame shift mutations. So intercalating agents are actually fairly common. They are wonderful stains of DNA. Okay, they're fluorescent stains. Like every DNA-based lab that's been around for more than 10 years has got a bottle of lithium bromide laying around. Okay, fantastic stain of DNA, fluoresces it bright pink, but known intercalating agent. So here, they have chosen to use acridine orange. Like every other book uses lithium bromide, but whatever. So acridine orange stains DNA. It does that by not just binding to DNA, but by slipping between the rungs of the DNA ladder. So it slips between and then it wedges itself. So it goes between the rungs and kind of bubbles. This deforms the DNA. Think of it like stretching that phosphate sugar backbone. And when DNA polymerase gets to that intercalating agent, it can add or remove nucleotides. It can lead to deletions or insertions. And that causes frame shift mutations, right? You add or remove nucleotides, you push the reading frame out of frame. All right, so any questions with the chemical mutagens? All right, let's tackle the physical mutagens. These tend to be pretty easy. So just radiation. So going down the electromagnetic spectrum, you got radio waves, microwaves, infrared, visible light. And then once you get past purple, you hit ultraviolet. Anything from ultraviolet on through gamma radiation is very high energy. And DNA will readily absorb the energy from anything from UV on. And when it absorbs that radiation, it will damage your DNA. Okay, so let's see what happens. All right, so ultraviolet radiation isn't too awful. It's non-ionizing, so it's not going to break your DNA. UV radiation is harmful because your DNA will absorb it and cause what's called thymine dimers. So let's see what a thymine dimer looks like. A, C, G, T, T, A, G. Made up sequence. I got to make sure I have adjacent thymines. T, G, C, A, T, C. All right, so this is what happens. Okay, your DNA absorbs UV radiation. Thymines, when they're next to each other, get particularly excited. And once these adjacent thymines absorb enough energy, they will form an extra set of covalent bonds. Double bond, double bond. Thymine dimer. So, prettier picture here, but the adjacent thymines form a new set of covalent bonds when they absorb enough UV light. So this deforms the phosphate sugar backbone. So when you step out in the sun and your cells are absorbing UV light, it's damaging your DNA. Okay, and this is what sunburns are. Sunburns aren't like your skin got burned. Sunburns are radiation burns. Your body says, well, those cells absorbed too much UV light. There's too many thymine dimers. We're just going to kill them. Okay, that's a sunburn. Fortunately, there is a way to repair these, which we'll get into, but thymine dimers can lead to all sorts of mutations, point mutations, frameshift mutations. So once they accumulate enough, it just kills the cell. Okay, this is why we throw our goggles into that UV cabinet at the end of lab. We cook any bacteria that came off your face by just killing them with thymine dimers. All right, now, regarding ionizing radiation, so X-rays and gamma rays, these have a lot more energy in them. And X-rays and gamma rays have enough energy to break the phosphate sugar backbone. Okay, X-rays tend to lead to single-strand breaks. Gamma rays will outright shatter DNA. Okay, this is why gamma radiation is becoming a very popular sterilant of imported goods to kill any fungus or lizards or whatever else decide to come along for the ride. All right, so this is also why X-ray techs have to monitor their yearly exposure, make sure they're not accumulating too much damage to their DNA. So, when you start breaking DNA into pieces, though, that's going to lead to mutations. All right, so that's our physical mutagens. You got UV radiation that causes thymine dimers, and then X-ray and gamma radiation, which start breaking DNA into pieces. Okay, now, this has been all doom and gloom, right? We talk about how mutations happen, how we can force more mutations. Let's talk about how we can fix them. So, there are many DNA repair systems. This is just some of them. Okay, so let's see some examples here of how you can fix these mistakes before they become a problem. All right, so first repair system occurs during replication. DNA polymerase proofreads its own work. Okay, this is why it only makes a mistake once in every billion nucleotides. It makes more, it just catches them before it leaves them behind. Now, what if it does leave a mistake behind? Okay, who's going to clean up that mess? That is the mismatch repair system. So imagine the mismatch repair system as a set of enzymes that are following right behind DNA polymerase, cruising down that DNA, cleaning up the mess. All right, so let me fix this here real quick. All right, so let's make a mistake here. Let's take our old example. Let's do an AC pair. All right, so here's our example sequence with one change. Instead of being AT or GC, this is now an AC pair. So in the mismatch repair system, we need to find these mistakes. Here's the problem, though. The mistake is only on the new strand, right? The old strand's fine. The old strand's template, so mistakes only start on the new strand. The first question is figuring out which strand is new, right? You're an enzyme, you got to sort out which one's which. I'm leaving out part of the story. Okay, there is a way to tell them apart. Right before DNA is copied, it gets methylated. So you get all these CH3s attached to the backbone. So right after replication, I don't know why I can't spell. The old strand is methylated. The new strand is not. Okay, it hasn't had time to be methylated yet. It's brand new. All right, so red strand is a new strand. We know that cytosine is the mistake. How do we fix it? Well, this is always entertaining to draw. And this is something I don't show you in most textbooks at this level. That's better. All right, so we draw two donuts and a horseshoe. This is what's called the MUT proteins. They are the enzymes involved in DNA or mismatch repair. So why the donut shape? They're like clamps. Okay, they're clamped onto the DNA, then they slide down the strands. So this guy's going to be sliding this way, looking for these mistakes. And when it finds a mistake, it fixes it. Okay, looks for the new strand, identifies a new strand, repairs it. So let's see how it gets repaired. I never remember the example sequence here. Sorry. Okay, so here in this example, I always mix up the methylated strands. The top strand is methylated. So that's the old strand. The bottom strand is a new strand. And here we've got a GT pair, right? This thymine's got to go. So how do we do that? Well, it's actually pretty easy. You cut the regions surrounding the mistake. So now we've got a gap. DNA polymerase comes back, fills in the gap with DNA, and then our old friend ligase shows up. Remember, ligase sealed up the Okazaki fragments, sealed up each of those lagging strand pieces. Here it does the same thing. It seals up this newly corrected DNA strand. Okay, so that's it. So these proteins find the mistake, they cut out the region surrounding the mistake, fill it in with the right DNA, seal it up. Now, let's talk about repairing thymine dimers, right? How is it that we can survive in the presence of ultraviolet light for some time before sunburns happen? You actually have two repair systems for fixing thymine dimers. One repair system for if the sun is out, the light repair system, and then another system for when it's dark out, the dark repair system. All right, so how does the light repair system work? So when you step out in the sun, thymine dimers begin to accumulate. most organisms make an enzyme called photolyase. Okay, so photo, light, lyase, to break or cut. So light cutters. So photolyase finds thymine dimers, breaks the covalent bond, flattening out the DNA. Now, the most ironic part about this, photolyase uses energy from the sun to do this. So, how weird, right? Light causes thymine dimers, light fixes thymine dimers. Now, if there's any lingering thymine dimers and it's dark out, there's a different repair system, the dark repair system. Now, the dark repair system works just like the mismatch system. So, it finds a thymine dimer, cuts out the region surrounding it, and then fills in the gap with the right DNA. Okay? And then ligase seals it up. All right, so that's fixing thymine dimers. Now, what about repairing damage from chemical mutagens? Say, nitrous acid exposure or base analogs. Okay, you have a repair system just for that. So, repairing modified bases or removing base analogs works the same way. So, a different set of enzymes shows up, glycosylases. They find the incorrect base, the modified base, and they actually will cut out the nitrogenous base. So here you can see I'll cut this backbone to start. But once they remove it, look what happens. Same series of events. Cut out the region surrounding the gap, fill in the space with correct DNA, seal it up. So with most repair systems, it's all the same. Find the mistake, cut out the mistake, fill in the right DNA, seal it up. Okay, it's just a difference of what is the mistake to start. All right. Any questions about those repair systems before I throw a curveball at you? Okay. One more repair system. I just basically want you to know what it's for. The SOS repair system. This repair system is in place to repair heavily damaged DNA. What do I mean by heavily damaged? I mean broken into pieces. So here we're going to try gluing Humpty Dumpty back together again when you don't have all the pieces. So let's imagine we've broken up our DNA into a bunch of little fragments. Okay, how do we glue them back together? Well, in this repair system, there's actually 30 different proteins and enzymes involved. One of them, though, is more important than the others. An SOS DNA polymerase, which goes by a different name. DNA polymerase 5. So we've seen 3. That's a really important one, right? That does the bulk of the copying during replication. We've seen DNA polymerase 1. That's the one that replaces the primers with DNA. And now I'm throwing 5 at you. So DNA polymerase 5 is a little different. Okay, it can copy DNA, but it does not proofread. So this is what it does. It takes the fragments and will just glue them back together, throwing in random nucleotides. So there's no proofreading, no double checking its work. It's just trying to glue all these broken pieces back together the best it can. So now your DNA is full of all sorts of new sequences that wasn't there before. That's gonna lead to mutation. So this seems pretty severe, but it's a last-ditch effort by the cell to survive. It's like my DNA is shattered, I got to glue it back together somehow. Okay, that's what this DNA polymerase is for. And then, fun fact, you would think that humans or some other eukaryote has the best DNA repair systems. That belongs to a group of bacteria in the genus Deinococcus. Okay, one, though, is king, Deinococcus radiodurans. We found it growing in a nuclear reactor. It can handle more radiation than any organism alive. And we figured out because it has the most efficient DNA repair systems on this planet. Its SOS repair system is basically in a constant state of repairing the DNA, and not without proofreading, accurately. Okay, we're actually worried that we have contaminated Mars, the Moon, and other celestial bodies with this organism, because we know it can survive cosmic radiation as well. So that's a fun one. All right, so all this talk about mutants and mutation, it's probably good to talk a little bit about how you can identify mutants. How can you tell when you have a mutation? So I've got two ways of doing that. So two types of selection tests. So you can identify mutants either through positive or direct selection, or you can identify them through negative or indirect selection. So what's the difference? In positive selection, you select for the mutant, not the wild type. In indirect selection or negative selection, it's the opposite. You select for the wild type and not the mutant. And by not having the mutant grow, you identify it. But I'll explain that with an example. So when we go through this, a couple of things. One, you do this by using selective media. And two, we're talking about this now because in the next chapter, we're going to talk about genetic engineering. And part of genetically modifying bacteria is knowing when you have a mutant and using the right test to identify those mutants. All right, so positive selection. The classic example here is identifying antibiotic-resistant mutants. So say you have a culture of bacteria that's not normally resistant to, say, streptomyces. But you suspect that in this culture you have some mutants that are all of a sudden resistant to that antibiotic. How do you identify them? You grow them on selective media. So here, we throw them on plates that contain streptomycin, the antibiotic in question. So at the plate on the left, if you get even a single colony, that's a mutant. That mutant is resistant to the antibiotic. Okay, the plate on the right, that's a control plate. No antibiotic, everything grows. Okay, so here, we select for the mutant. Now, in negative or indirect selection, it's a little different. Here, we're going to identify the mutant because it doesn't grow. So, the classic example here is identifying auxotrophic mutants. You're like, all right, I vaguely remember that term from the last test, right? Auxotroph versus prototroph. Auxotrophs need supplements. They need growth factors to grow. Okay, so in this example, we're trying to identify histidine auxotrophs. So bacteria that need histidine. So in this example, we start with a Petri plate with colonies. Okay, this media contains histidine. All of the colonies grow. So what we're going to do is we're going to transfer the colonies using sterile velvet. Yes, velvet. You press that velvet against the plate, and you get an imprint of all the colonies. And then what do? You take the velvet and you start pressing it to clean plates to inoculate. So we call this replica plating because you're replicating the first plate. All right, so here we press the plate on a plate without histidine and a plate with histidine. Okay, you incubate, you come back, and you compare the plates. Now remember, this is indirect selection. So, checking the plate without the histidine, it seems to be missing a colony. That's your mutant. It couldn't grow without histidine, whereas the others could. So that's your auxotroph. Okay, you check the control plate. Hey, look, the colony's back. So you grab the mutant, you put it on a new plate, now you've isolated it. Okay, so here we identify the mutant because it doesn't grow compared to the wild type. So you indirectly identify it. All right, so any questions with the selection tests? Because we'll see more of it in the next chapter. Haven. Wait, let me go back up. What's that?
Why must selective feature be used again?
Because you want to select for the mutant in some way. You're trying to tell the mutant apart from the original wild type. So there's got to be some way to sort that out. Selective B is usually a go-to option, so that you kill one of the two. Okay, so changing gears here, getting away from just plain old mutation. The next part we're going to look at is something that tends to be new to people who've taken this class for the first time: horizontal gene transfer. Okay, this is something they tend to skip over in Gen Bio. So horizontal gene transfer is something pretty common to bacteria, but we're finding can happen between any organisms. But here, we take two organisms, so two bacteria, and we trade DNA. So we move DNA between the two organisms. So horizontal, between the cells, gene transfer, moving DNA. So let me start diagramming this here. So two things must happen, two criteria must be in place for horizontal gene transfer to occur. First thing is you need a donor cell. Second thing is you need a recipient cell, because we're going to move DNA between donor and recipient. All right, now we need to draw some DNA here. Okay, so here's the donor's chromosome, and we need to draw in plasmids. So it's been a while since we talked about plasmids. Plasmids are extra chromosomal pieces of DNA. Okay, bonus DNA, little mini chromosomes. And they are very commonly transferred in horizontal gene transfer. All right, let's give the recipient cell here some blue DNA. Okay, so that's its chromosome. There you go. All right, so you have to have a donor cell and a recipient cell. That's one. Two is DNA must be transferred. So plasmids. Plasmids are very commonly transferred in horizontal gene transfer. So the donor can give copies of its plasmids to the recipient cell. Now this is common because plasmids take care of themselves. They copy themselves, they're autonomous. So if the plasmid gets transferred successfully, it becomes a part of the new recipient cell, like a new little pet. Now where things get tricky though, transferring fragments of DNA. Because that's a possibility. A fragment of the donor chromosome could potentially be transferred. Here's the problem, though. Fragments of DNA do not take care of themselves. Okay, they have to be stabilized in some way, because otherwise enzymes are going to eat them up. So we have to think, how do we stabilize a fragment of DNA? Well, we're going to put it into the chromosome of the recipient. Just patch it in like it's always been there. So that process is called recombination. It's very complicated. We'll touch on it superficially. But here we literally recombine DNA. So DNA from two different organisms recombined, glued together. Okay, so you have a donor and a recipient. If plasmids are transferred, they're fine. They take care of themselves. But if fragments of DNA are transferred, they must be patched into the recipient chromosome. Otherwise, they are destroyed. All right, so we'll touch on the first one here, but there are three mechanisms of horizontal gene transfer. Three ways to move DNA from donor to recipient. We're going to talk about transformation here momentarily, where the donor is dead and we just accept naked DNA. Oh, I have pretty pictures this semester. I forgot. So transformation is the uptake of naked DNA by the recipient. Transduction is transferring the DNA using a virus as a vector. And then conjugation is where things get a little bit more intimate. The cells make contact and they directly exchange DNA. All right, so going back to that fragment of DNA there, and I said that somehow it gets recombined into the recipient chromosome. And I mentioned that's through a process called recombination, or genetic recombination. This is something you don't get into much until you take molecular biology. And you find out there's several different types of recombination, and they all suck. Okay, they're all very complicated. But one example, keeping it as simple as possible, is homologous recombination. Okay, so homologous. Okay, that's talking about things that are similar to each other. They can be identical, but at the very least, they must be similar. So here, this is the swapping of DNA, the exchange of DNA between segments of DNA that are similar to each other. They are homologous. So this example actually happens during meiosis in humans. All right, so in the example, you have two chromosomes, A and B. Chromosome A and B have similar genes, E, F, G, H, I. Now, these genes are similar to each other, but a little bit different. Here they illustrate that with upper versus lowercase, right? Lowercase E through I versus uppercase E through I. Because these segments of DNA are similar, they can align. And then we're going to physically cross them over. So in the middle diagram there, you can see the DNA has been physically crossed over. There are enzymes that do this we're not going to talk about. And fun fact, that crossing over there, it's called a holiday junction. But anyways, the strands get crossed over, and then they get resolved. And then look at the outcome. So, the top chromosome is lowercase e, f, g, but look at the ends. Now it's uppercase H and I. Okay? The bottom strand is opposite, right? E, F, G uppercase versus lowercase H and I. The ends got swapped. This is similar to how a fragment of DNA can be put into a chromosome. Okay, if it matches up to a portion of that chromosome in any way, it can swap in, switch places. Okay, so get this done here. All right, so the first example of horizontal gene transfer I have for you is transformation. Okay, this is pretty common in the bacterial world, but this is the uptake of naked DNA fragments. Now let's talk about how the DNA fragments become naked. In transformation, typically you start with a dead donor cell. All right, so when bacteria die, they decay. Their DNA begins to break down. So what are you left with? A pool of plasmids and fragments of DNA. The recipient cell may see this as an opportunity. It says, hey, those plasmids look useful, or hey, those fragments of DNA have useful genes. I want them. And it wants to bring that DNA into its cell. But there's a problem. Okay, fragments of DNA are big. And as we saw 2 weeks ago in lab, to get macromolecules in the cell, they got to get chopped up. But we don't want to do that. We don't want to chop them up. That would ruin them. So there is a way around that problem. The recipient cell will open up its cell wall a bit. It becomes, as we call it, competent. So a competent bacteria is willing to accept DNA. It's willing to be transformed. So the recipient cell alters its physiology, alters its cell wall, so that the fragments of DNA or the plasmids can enter. Now, not all bacteria like being competent. Typically, gram-positive bacteria love being competent. Okay, gram-negative bacteria, not so much. But I'll come back to that in a bit. But otherwise, if a plasmid enters, it takes care of itself. If a fragment of DNA enters, it's got to be patched into the chromosome. It's got to be recombined. All right, so quick experiment just to illustrate this. So this was first witnessed by scientists in 1928 by Frederick Griffiths. So Frederick Griffiths was trying to develop a vaccine against streptococcal pneumonia. Okay, so let's take a look at how his experiment worked here. So there are two types of strep pneumoniae. There's rough type and smooth type. The rough type is non-virulent. The smooth type is virulent. So why is it rough or smooth? It's all about having a capsule. Okay, smooth streptococcal pneumoniae has a capsule. This is why it's pathogenic. It needs that capsule to stick to your lung tissues. The rough type is non-pathogenic because it doesn't have a capsule. Thus why it's rough. All right, so in the control experiment, he injected a mouse with the rough type. Okay, rough type goes in, poor little mouse dies. Now, in the first experiment, he's like, maybe if I cook the bacteria and turn it into soup, that debris would serve as a good vaccine. So he took the smooth type, he cooked it, turned it into debris, and injected it in that happy little mouse. Mouse was fine, but it wasn't a good vaccine. So he's like, okay, let me try this again. I'm going to mix together living rough type with dead smooth type. So you take the living rough cells, mixed together with the soup of the dead smooth cells. So I thought, okay, it's a perfect cocktail, right? I got everything I need. I got some living cells that are weak, and I got some of the debris from the virulent cells. Well, he put that cocktail into the mouse, and look at what happened to that poor adorable mouse. It died. So he's like, oh God, what happened? Why did the mouse die? It shouldn't have died. So he removed a sample. And what did he find? From the mouse, out came nothing but smooth type. So rough went in, smooth came out. So he took these new smooth cells, injected into another poor little mouse, and that poor little mouse died. So what he witnessed here was transformation. This is what happened. Say this is a gene for a capsule. The capsule gene was acquired by the rough type. It integrated that capsule gene, and guess what it can make now? Capsules. So it got transformed from being rough into smooth type by accepting DNA from the dead cells. Okay, we had no idea that was happening at the time, but now we understand. All right, so any questions with that stuff from last time before we move on? All right, so the second mechanism, I'll tell you here and now, I can't draw. It's a little bit more complicated than I'm capable of, but transduction. Transduction is the movement of DNA between bacteria by a virus. So a virus is going to serve as what's called a vector, okay? An entity that moves something between organisms. Here, it's going to move DNA from one bacteria to another. It's like a DNA shuttle, if you will. So a little hand waving here because we'll get to this in a chapter and a half here, but we're going to get to the virus chapter soon, which one I find a lot of fun and very interesting, but the emphasis on that chapter, as we handle it in this class, is bacteriophages. Okay, so a bacteriophage is a virus that infects only bacteria. Okay, there's viruses for all organisms. Nobody's safe. But there are viruses exclusively for bacteria. So bacteriophages are going to be the vectors here. They're going to be moving DNA between organisms. And that, again, is transduction. So we're going to talk about this more when we get to the virus chapter, but there are two types of transduction. Okay, generalized, which I will talk about today, is the movement of any DNA, any genes from the donor cell to the recipient cell. That's why it's generalized. Any piece of DNA in that donor's fair game to be moved. Specialized transduction, I'm not going to touch until we get to this virus chapter coming up because there's more involved. Okay, there's more rules. So with specialized transduction, only specific genes, specific DNA may be transferred. All right, so just a kind of a heads up on what's coming up. This is going to be the main focus in the virus chapter. We're going to spend a lot of time talking about how viruses replicate. Okay, so what happens when a virus infects your cell? So there will be two replication cycles we'll get into. I just want to focus on the top, the lytic cycle. And this is the simplest one I can find. That's why I use this old diagram, but just the basics. So viruses are obligate parasites. Okay? They need to infect living cells to replicate. So the first thing that happens during infection is that the bacteriophage here, the virus, attaches to the appropriate host. It then injects its DNA into the host cell, like an evil little syringe. And then it takes over the host cell. Okay, guts out the host cell, breaking up its components, and it then forces the host to make more viral components. So it turns the host cell into a factory for viruses. As time passes, the host cell is then forced to assemble the new viruses until the inevitable. The viruses burst out, killing the host. So this is a cycle any virus is capable of. So when you got a cold, this is what they're doing to you. They're getting in your cells, they're taking them over to make more viruses, killing that cell in the end. So why does this apply to transduction? Because nothing in nature is perfect. Mistakes happen. And transduction is a mistake during the lytic cycle. All right, so different diagram here, similar story though. So here the bacteriophage, the virus, finds the appropriate host cell. Right? It injects its DNA, and then it takes over that host cell. Now, notice what it does to the host chromosome here. It chops it into itty-bitty pieces. Okay, it's to take away the host defense. So it cuts up the host chromosome. Now, this is where the mistake occurs. Typically, viruses are assembled very carefully so that when viruses are put together, they're only packed with viral DNA, right? It only makes sense, right? The virus should get viral DNA. Once in a great while, you'll get the oddball dud. So the guy there with purple DNA has bacterial DNA in it. Okay, that's a mistake. Right? It shouldn't have bacterial DNA packed in it. It should have viral DNA. But it happens. So the new viruses get out, right? They punch out. This dud gets out. But look, it can still attach to host cells. So if this is your donor, that's your recipient. So the recipient cell's infected by this dud virus. It injects that purple DNA. And then what happens? Recombination, if it's lucky. So here, the virus is going to transfer DNA from the donor accidentally. And the virus gets packed with the wrong DNA. It goes out, infects the recipient cell, injecting it with the wrong DNA, with bacterial DNA. And then if it recombines, that's horizontal gene transfer. Okay, now this is generalized because any piece of that donor DNA, in theory, could have been packed. They're all fair game. Okay, specialized transduction, when we get to it, we'll have more rules to it. So only very specific DNA can get transferred. All right, so that's transduction in a nutshell. Any questions on how that works, especially where the virus goes wrong? Okay. All right, last mechanism here I can draw. Conjugation. This is also pretty common, and it's starting to worry us, which you'll see why. So in conjugation, the donor and the recipient make direct contact. Now, they make direct contact through something we talked about long, long ago. Sex pili. That's way back in chapter 3. We talked about cell structure. We talked about pili. There was fimbriae, which are those short hair-like guys used for attachment. And then there was sex pili, long, hollow tubes used to connect bacteria, okay, like a docking station. So the point of the sex pili is to allow DNA to pass through. So the donor can conveniently ship DNA through that sex pili to the recipient cell. Now, folks, not all bacteria can do this. Okay, pili are protein. Protein are the product of genes. So the only bacteria that can do this must have the genes for pili. They have to have the genes to initiate this process. Okay, and that's going to be an important thing to understand. Okay, and you'll see why, but, okay, donor has the genes to do it, connects with the recipient, sends it DNA. Now, what's sobering about this is the fact that this is really the only form of horizontal gene transfer where everybody lives. Because look at transformation. That DNA was from a dead cell, right? A dead donor. Look at transduction. We just said the virus kills the host, the donor, in the process. But in this conjugation, everybody wins. Come together, they make their exchange, they leave. Oh, and before I forget, typically plasmids are transferred in conjugation, but technically speaking, fragments of DNA could too. But I don't get into that model anymore. Okay, so when talking about conjugation, knowing that the donor has to initiate this, you often assign mating types, kind of like male and female. Okay, here you figure out who's the donor, who's the recipient. So the classic example of mating types involves a specific plasmid called the F plasmid, or fertility plasmid. All right, so why does this F plasmid matter? Why is it called the fertility plasmid? This plasmid carries the genes for conjugation. The plasmid itself has the genes to make the sex pili. So, if a cell has this plasmid, can it initiate conjugation? Yeah. If it doesn't have that fertility plasmid, can it initiate? No. Okay, that's why they're mating types. Okay, the donor with the plasmid to start we call F+. The recipient, who doesn't have it yet, we call F-. All right, so here's the scheme. So looking at the image, it's pretty easy to figure out who's the guilty party. The cell on the left that they've labeled F+, it's got Fimbriae. It's got the other type of pili. That's usually the giveaway here. So here you can see they make direct contact via that sex pilus. The plasmid's being mobilized. Okay, a copy is being sent over to the recipient. Now, here's a scary thing. Once the recipient gets the F plasmid, does it stay F-? It becomes F+. And then guess what happens after that? They go out, they find more cells to convert. So it's just this cascade of indoctrination, spreading the good word of the F plasmid, right? They just keep passing it from cell to cell to cell. Okay, there's going to be consequences for that, which I'll get into in just a second. Yay for OpenStax having a simpler version of this than the Pearson text. All right, so They make direct contact. Okay, there's your sex pills. Albeit that one's super duper short because, I mean, look at this beast. Okay, now what's interesting here is how the plasmid is transferred. So notice how it looks like it's peeling away the outer strand. So you remember how awful it was learning about DNA replication with leading strand, lagging strand? I got bad news. That's not the only type of DNA replication out there. This is called, you don't have to know this, rolling circle replication, which has got its own mechanisms. But regardless, a copy of the plasmid is sent over. They both become F+. And I do have an animation tucked away here. I got to update the link, so if it doesn't work for you, I'm sorry. I'll try to get the correct YouTube link. Can't remember which ones I fixed and which ones I didn't. So this is scary because everybody wins. These plasmids, as we are learning, often don't just carry pili genes. They're known to also carry antibiotic resistance genes. So we hear about in the news the growing antibiotic resistance problem. Right? More bacteria are learning how to be resistant to newer antibiotics. This is part of how they do it. They go out, they share this plasmid, and they say, hey, now you can conjugate, and now you've got tetracycline resistance. Okay? And they just keep spreading it. So on that note, and honestly, I should probably talk about these earlier, just, I don't know where to fit it. So just touching on plasmids again. Okay, plasmids are a common theme in this half of the chapter, right? In all three forms of horizontal gene transfer, technically plasmids could be transferred. Sometimes they are more often than fragments. So plasmids are, in my opinion, a really interesting topic. For one, people don't realize that there are plasmids found in all three domains of life. There are eukaryotes with plasmids, there are archaeans with plasmids, and there sure enough is bacteria with plasmids. Now, the most fascinating thing about plasmids is that while typically they do carry genes, sometimes a few, sometimes hundreds, they almost never encode for essential traits. So the genes that plasmids come with are non-essential. So you could rip them out of the bacteria, and the bacteria would be just fine. So what kind of traits are non-essential? What would you find on a plasmid? Well, we just mentioned that many plasmids carry antibiotic resistance genes. Okay? Pili synthesis for conjugation. Here's a fun one. Toxin production. There's more bacterial pathogens out there than most folks realize that produce toxins from a plasmid. Okay? Cholera does this. Anthrax does this. Okay, diphtheria does this. So they produce nasty toxins because they have a plasmid that allows them to do so. And the weirdest one, my favorite here, oddly specific, tumor formation in plants. If you ever take a full-fledged biotech class in the future, they talk about a bacteria called Agrobacterium tumefaciens. This guy's weird. It undergoes conjugation, not with other bacteria, with plants. So a bacteria makes a specialized pili with plants, forcing plasmids into the plant cell. It seems really weird, right? Why would it want to give a bacterial plasmid to a plant? The plasmid causes the plant cells to form tumors. Why? Because what is a tumor? It's an overcharged cell, metabolically unchecked. And for plants, that means sugar, and lots of it. So the bacteria causes tumors to form in the plant just so it can have the excess sugar that it makes. That's pretty weird. But we exploit that for genetically modifying plants. Other neat things about plasmids. You know, when you draw them, you know, you can draw like one plasmid or maybe a few. Some are, as we call it, low copy number. You know, there's only a few copies per cell, maybe just one. Many plasmids, though, are high copy number. Thousands of copies per cell. And I bring this up because we'll talk about that a bit more in the next chapter. And then another thing, right? I make it look like these plasmids can be transferred between any bacteria. Not all of them can. Some plasmids are narrow host range. They can only be transferred between specific bacteria. Just gram-positives, just gram-negatives. But the scary ones are broad host range. They can be passed around to anybody. So that with antibiotic resistance, okay, this is a big problem. Or a real problem. And then this, I'm just going to skip. I don't have any questions about it. This is just an example of like a resistance plasmid. This is what we're worried about now. So not only is this plasmid capable of conjugation, it also comes with resistance to 3 last resort antibiotics. So this one here doesn't exist as far as I know, but if it does, we'd all be boned. All right.